Okay, so thank you so much for the invitation to talk about tumor sequencing and tumor heterogeneity, which is the topic of the research in my lab. So this is how we think about cancer in general in my lab. We think about cancer as a Darwinian process. We have this kind of picture in mind all the time. Basically, tumors and cancers as a Darwinian process where you have a single cell that acquires a mutation that makes the cell sort of divide and expand its sort of abundance. And basically what's happening is that each cell is independent. Each cell can acquire mutations individually, and each cell can expand its abundance in the population. And this is all in the context of selective pressures that are coming from multiple sources, the microenvironment, the cells themselves, and obviously treatment as the main source of selective pressures. So we're trying to understand how tumors evolve in general. And we use the tools of high-throughput sequencing as a way to do so. So essentially using machines that can sequence DNA, a very large amount of DNA in a very little amount of time. We do focus on B-cell lymphomas. And so the topic of this lecture is to try to understand how B-cell lymphomas evolve upon treatment. So as you all know, B-cell lymphomas are coming from B-cells, and B-cells are very special in the sense that early on in the process of B-cell development, B-cells undergo a process called VDJ recombination. This is a process where there is a single V segment from the germline genome that's picked randomly from a group of Vs, a single D segment that's picked randomly as well, and a single J segment that's picked as well randomly. And these three segments picked randomly are fused into what's called a VDJ recombination. This is what codes for antibodies, as you all know. Later on, these B cells, which basically tag with a unique tag, each B cell is a unique rearrangement, these B cells enter the germinal center, and they undergo another sort of layer of diversification of the VDJs, where there is a protein called AID that comes into the VDJ and makes point mutation into the VDJ junction. The goal is to generate better antibodies, more heterogeneity in terms of the immune response and so on. Now, as many of you may know, this mutational process is leaky. AID is also making point mutations elsewhere in the genome, and that contributes to leading to lymphomas via mutations of oncogenes and tumor suppressors. But what's happening is, and so that leads to essentially B-cell lymphomas. But what's happening is that after the B cell becomes a lymphoma cell, this process, this mutational process here, keeps going for a little while. And what that means is that if you have a B cell lymphomas with many, many cells, this protein that makes point mutation into a VDJ is acting in every single cell, right? And so what that leads to in the context of a population of tumor cells is essentially heterogeneity in terms of the mutational patterns of the somatic hypermutations onto the VDJ junction. This is what we use as a way to study clonal heterogeneity in these tumors. And again, we use sequencing as a way to do so. So we essentially sequence these VDJ junctions. They are in the order of 300 base pair long. So we can actually now sequence the entire VDJ junction using modern sequences that can sequence reasonably long fragments of DNA. In fact, the latest sequences can sequence about 300 base pair long fragments. The advantage, again, is that we can sequence millions of them in the same run. And so so that's what we do. We sequence millions of these VDJ junctions per tumor, and we look at the extent to which these junctions differ from each other in terms of sequence patterns. So we apply this technique, this high-throughput sequencing technique, to B-cell lymphoma patients that are treated at Clonal at the New York Hospital. And so we have biopsies, essentially, at multiple time points, typically diagnosis biopsies and relapse biopsies. And we try to understand how heterogeneity changes in time between these two time points, sort of given the fact that these patients are being treated with standard chemotherapy between the two time points. So when we sequence VDJ junctions from a given biopsy, from a given tumor, this is what we see. This is the type of pattern that we see. What we do is very simple. We just count how many times we see each combination of VD and J, right? And what we see in a given tumor is always like this. We always see a very sort of major abundant clone. This is the same VDJ. This is the identity of a VDJ here. And basically, this is consistent with the clonal origin of a tumor. There's one B cell that gave rise to the lymphoma, and all of the B cells share the same VDJ. This is just showing you the identity of a VD and J. This is not making any sort of statement yet about the mutational patterns onto the VDJ. So this is what we see in each individual tumor. We also see, as you can see here, the smaller clones of other B cells, basically, and we think that these are infiltrating B cells, essentially B cells that find their way into the tumors and that we sequence in the process of the sequencing the VDJs for the whole biopsy. Now, when we apply this technique to multiple patients, one thing that we can do is to look at this major clone that I showed you. We can look at the identity of a clone, and we can look at the identity of a clone at diagnosis and relapse and try to compare these identities. And in almost all cases that we sequence, we see the same VDJ at diagnosis and at relapse. Okay? So basically, it tells you that the disease that comes at relapse is the same as the one that you saw at diagnosis. And it's actually striking that sometimes relapse is happening many, many years after the initial diagnosis. For example, here, this is a patient where the relapse came 11 years after diagnosis. But we can tell that it's the same tumor because the tumors have the same VD and J, right? Very striking that the tumor was essentially gone or essentially sort of dormant for so many years and came back 11 years later. There's only one case where the tumor that came back 6 years after the original diagnosis was a different tumor based on this VDJ pattern. So this was telling you something about VD and Js. This is here looking at the mutational patterns of the major VDJ clone that we see. This is each segment here corresponds to a mutation pattern. This here tells you something about the abundance of a mutation pattern in the clone. As you can see, there is one mutation pattern that's very abundant, and you have other mutational patterns that are not too different but have lower abundance. You also see that you have mutational patterns that are significantly different, but at the low abundance in this particular patient. This is again one time point, one patient, and the main VDJ. So we do this counting process at diagnosis. We do the same thing at relapse, and we also see similar pattern, a very, very abundant clone, but also sort of smaller clones that are different in terms of mutational pattern. Each of those little vertical lines corresponds to a different mutation, a different AID-induced mutation. Okay. But it's, again, this is the VDJ at diagnosis and relapse is the same. So we can put everything together, essentially, and to sort of put everything together, what we do is clustering, or we actually essentially build phylogenies of tumors. We essentially try to identify the similarities between these segments, so putting both diagnosis and relapse in the same tree. And this is what this is the kind of pattern that we find when we do this phylogenetic analysis of these tumors. This is a case here where all these figures are going to be the same. What you see in blue are VDJ sequences that we see at diagnosis. What you see in red are VDJ sequences we see at relapse. Okay, so blue is diagnosis, red is relapse. These are mutational patterns, same thing that I was showing you in the previous slide. And this is the tree of these mutational patterns. The size of a bar here tells you something about the abundance of a particular pattern within the tumors, within the biopsies. And so what you can see here in this particular patient is essentially a situation where the relapse clones are growing out of the diagnosis clones, right? There's one diagnosis sort of cell or clone that's picked probably randomly or for certain reasons. due to the sort of probably the mutational patterns in the rest of the genome. And essentially that was picked up and expanded its abundance in the relapse tumor, gives rise to the relapse tumor essentially through this evolutionary process. So this is a situation that we see here. We see this kind of situation quite a bit too. This is a situation where you can't really tell the difference between the VDJ junctions that you see at diagnosis and relapse. They all look very similar to each other. We do think, and I'll show you some other data that shows that it's likely to be the case, but this is also a type of scenario where it's a linear scenario in terms of evolution, where basically you have a major diagnosis clone. There's one cell that's picked up and gives rise to the relapse tumor. But we see more interesting scenarios that are nonlinear by nature, and I'll show you two of these. So this is a patient here that was diagnosed with diffuse large B-cell lymphomas in 2004, treated with CHOP, and a relapse with a low-grade B-cell lymphoma in 2006. So we applied this technique to this particular patient. And here what we see is a significantly different scenario, where instead of seeing this kind of linear evolutionary scenario, now you have essentially a divergent, a parallel evolutionary scenario, where essentially the relapse clones and the diagnosis clones are clustering into two different branches of the same tree, right? Again, this is the blue is diagnosis, red is relapse. And what you see is that so they essentially evolved across different branches, across different paths, but they do share some similarity. They share a common ancestor. And obviously we are very interested in understanding what happened at this branching point here. Essentially something happened, gave rise to a cell that was able to regenerate two tumors independently across two different paths. The diagnosis tumor came early, the relapse tumor came later, but essentially these two tumors have the same origin. What's very interesting is that we did find a tiny clone. We're talking about 10 VDJ sequences out of 500,000. This is the kind of numbers that we have in this type of analysis. But this little clone that we saw at diagnosis is actually not clustering with the rest of the diagnosis clones. It's clustering with the relapse clones. Basically, this is something that we see at diagnosis. Right? Very, very tiny clone, divergent, different mutational pattern. This little tiny clone is actually not killed by the chemo. The main diagnosis clone is killed, essentially not found at relapse. But this little diagnosis clone that's essentially survived the chemo is able essentially to regenerate the entire relapse tumor. So something very similar to the concept of stem cell, able to withstand chemotherapy and also regenerate entire tumors. And we are doing a lot more research trying to understand exactly the nature of this kind of small clones. This is another patient with a similar type of scenario. We had two biopsies at relapse in this particular case, so the tree is a bit more complicated, a bit more interesting. But this is the same type of picture where, as you can see, there's essentially parallel evolution of diagnosis and relapse and relapse tumors. They came from the same ancestor, but they diverged early in terms of evolution. And as you can see, there's a pattern of evolution in terms of relapse that seems to indicate that one side of relapse was able to seed another side of relapse, as you can see in this tree here. Right? So this is only looking at the VDJ junction itself. It's a very, very small chunk of a genome. We are obviously interested in what else is happening in the genome. And so that's why when we want to sequence more, we want to sequence almost entire genomes. We don't sequence entire genomes for practical reasons. We sequence all of the genes in B-cell lymphoma genomes using a process called exome capture and sequencing. We essentially sequence all coding regions of these tumors. And that tells you something. We are essentially looking for mutations that happen somewhere else in these tumors. And what we do is to, likewise, to compare the diagnosis and relapse. We try to understand what are the new mutations that came up at relapse that were not there at diagnosis, potentially identify mutations that went away at relapse compared to diagnosis. But in this kind of patient here, pretty much systematically, where I was showing you this kind of mixed pattern of VDJ sort of phylogenies, and I was telling you that it's likely a linear scenario, we see this kind of pattern here looking at abundance of mutations between diagnosis and relapse. We see a lot of mutations that have conserved, sort of maintained their abundance between the two time points. but we see a lot of new mutations in relapse compared to diagnosis. Again, this is consistent with this kind of linear evolutionary scenario where there's additional mutations that are made onto one of the cells that's coming from a major clonal diagnosis. Some of these mutations are very relevant to B-cell lymphoma biology. For example, EZH2 mutation that came up wasn't there in the primary tumor that essentially appeared later on at a later time point in this particular tumor. This is just another patient with the same type of scenario. You see a lot of new mutations came up that were not there at diagnosis, came up in relapse. Also consistent with this linear evolutionary scenario. Now, the nice thing is, when you go back to these patients that had a more interesting evolution scenario, divergent type of scenario, we can actually now look at the mutational pattern in the genome, the rest of the genome, in the context of this phylogeny that we were able to draw based on VDJ sequences. Remember that this is a case here where you had this is patient 2, you had two clusters, two essentially sort of tumors that evolved according to, essentially in parallel path. So we have this phylogeny here. We can look at mutations that sort of came up in these tumors in the context of these phylogeny. As you can expect, we do see relapse-specific mutations, right? You would predict based on these kind of phylogeny that there will be diagnosis-specific mutations. Again, these two tumors evolved independently, so diagnosis, would have evolved its own mutations. And indeed, we see a lot of diagnosis-specific mutations. What's interesting about these tumors, I told you that the relapse was actually indolent. The diagnosis was actually more aggressive. This is actually, it fully makes sense in the context of these mutational patterns that we see here, right? These are very, very aggressive mutations, MYD88, B2M, these are ABC type mutations. The mutations that we see in this relapse tumor, again, acquired independently in a different branch, are actually much milder and more of a GCB type mutations. So the mutation is sort of pattern sort of explains the phenotypes of these tumors. Now again, I was telling you about this branching point between the tumors. What happened here. We're very interested in what happened here because something happened here again that was able to essentially make cells sort of pro-lymphoma, lymphomagenic. Essentially, mutations probably happened that were able to give rise to a cell that was able to originate two tumors independently. This is not something that's easily achievable in a sense. So the nice thing about doing this is that doing this kind of analysis, we can triangulate. We can essentially ask, what happened at this branching point in the rest of the genome. We can look for mutations that are common essentially between these two tumors and not find the germline. And we see this is the kind of mutation that we see. We're very excited about this mutation here, MLL2. This is a very, very frequently mutated gene in B-cell lymphomas. And this is, as you can see, this is a mutation that's shared between the two tumors, and we did additional sequencing to show that it's not in the germline. So really something that happened at this branching point here and potentially pushed a cell towards a pro-lymphoma state. Along these two branches, the cells had to acquire additional mutations to really become full-blown lymphomas. But we think that this is a very, very important initiating event. This is compatible with the very high abundance or very high frequency of the mutation in this gene in B-cell lymphomas. This is the same type of analysis here. Again, we identify relapse-specific mutations, mutations that are specific to one side of relapse, common to the two sides, diagnosis-specific mutations, and also mutations that potentially have appeared at this branching point here. Interestingly, in this patient, we also see a mutation MLL2, this chromatin-modifying enzyme that's highly frequently mutated in B-cell lymphomas. So this is, just a few more slides. I mean, to sort of to conclude on a slightly different topic, but very complementary. So we can use this information as a way to predict whether clonal heterogeneity diagnosis predicts the occurrence of relapse. Some patients relapse in B-cell lymphomas, some patients don't. We can ask whether clonal heterogeneity measured using VDJ sort of pattern differences predicts relapse. And it turns out not to be the case, at least in our hands. We don't see any difference in terms of sort of probability of relapse between patients that have a sort of a high, so the patients essentially who relapse and patients who don't relapse have the same level of clonal heterogeneity. So we're interested in looking at other things. And we looked at DNA methylation. This is something we've been doing for a while. Otherwise, we also use sequencing as a way to study DNA methylation. This is something that Ari mentioned in his talk earlier. This is sort of epigenome, looking at the epigenome. So we can use high-throughput sequencing as a way to measure, to quantify the epigenome. That's what we did in a subset of our patients. Also looked at normal B cells. And when we do this analysis of DNA methylation in this patient, we recover what other people have recovered. We see increased DNA methylation at CpG-islands. These are very highly CpG-rich regions of the human genome. Increased methylation at CpG-islands is something that you see in B-cell lymphomas. You also see that in many different tumor types. When you look outside of CpG-islands, you tend to see a small decrease in methylation. This is also something that's been seen by others. But the nice thing about using sequencing to study DNA methylation is that you can do other things, other analysis. And one very exciting analysis is to look at methylation heterogeneity. The extent to which, when you sequence methylation from a population of cells, all of those cells agree with each other in terms of methylation patterns, which is something like this, or essentially each cell has a different methylation pattern. So we can quantify methylation heterogeneity genome-wide using sequencing and using analysis. This is what we do when we compare diagnosis and relapse in terms of methylation heterogeneity within the same tumor. We see this sort of almost systematic decrease in methylation heterogeneity. It's consistent again with this sort of the clonal structure of these tumors, where there's one cell, a diagnosis that's picked up and likely gives rise to relapse, and therefore there's a decrease in heterogeneity. But what's interesting and very preliminary, but still, I figured I would show these results just because we're pretty excited about this, is that when we compare the methylation heterogeneity genome-wide between patients who relapse and patients who have not relapsed several years after treatment, we actually see a small but a significant essentially decrease in methylation heterogeneity in patients that don't relapse compared to patients who relapse. And basically, a way to interpret this is like this. Again, this is going back to this Darwinian process, that I was telling you about, not from a genetic point of view, but more from an epigenetic point of view. Well, basically, if you have a very sort of diverse population of cells, where each cell is a little bit different when it comes to its epigenome, it seems that you could have a slightly higher probability of relapse compared to if all of the cells are very similar to each other. And again, this is very much compatible with this Darwinian process where heterogeneity, diversity, makes it more likely that one cell has acquired something good that is essentially able to fuel the Darwinian process and essentially lead to a relapse phenotype. This is ongoing work in my lab, in my group. But these are results we're excited to pursue further and to study further. So this is the end of the talk. All of what I talked about today is a collaboration between my group and Wayne Tam's group at Cornell, who's the moderator of the session. We have a wonderful group at Cornell, very collaborative group. And so Peter Martin, John Lennon, Ari Melnick, Leandro Cajeti, very sort of, we talk all the time. We have a very sort of stimulating intellectual collaboration. And what I talked about today is a result of all this collaborative effort. So, thanks.
Thank you very much, Eric.